Hello dear aspirants, very good evening all of you. Welcome back to GDC Classes English, especially for your favorite series, that is Thunderstorm series, where you have a thunder-like impact on the specific topic. Yes, so now in this today's session, I'm going to discuss about cardiac glycosides, which are very important secondary metabolite right after the alkaloids. Yes, after the alkaloids. So the glycosides takes an important role in the dosage form preparation and especially for the therapeutic action. Yes, so without any delay, let's start today's session. But what you need to do is that please do share this video to the needful, and especially thank you for supporting us and thank you for watching the video. Okay, and yes, so let's start the today's session, cardiac glycosides or steroidal glycosides. But before that, so in order to understand the cardiac glycosides, first of all, we should know what is glycoside. What is glycosides? We know already the glycosides are the secondary metabolites and these are the organic compounds. But dear aspirants, okay, first and foremostly, when it comes to the competitive examination, GPAT, 𝙂𝙋𝘼𝙏, DI, Pharmacist, okay, or the state DIs or central DIs, okay. So when it comes to the Pharmacognosy, okay, so 60% of the syllabus will be comprised or which will covers the drugs part. In that, first place takes by the alkaloids, and second most important drugs classification is glycosides. Okay, yes. So now let us see in this glycosides classification. Okay, in the glycosides classification, we have three types of classification. First one is classification based on the pharmacological action. Classification based on the pharmacological action. Which action they were exhibiting? Either their purgatives, laxative, or cardiotonics, okay, or miscellaneous are there, right? So followed by that's one of the first classification. Second classification is a chemical classification. Okay, chemical classification, which means based on the chemical moiety, based on the 𝘂𝙀𝙎𝑃 chemical moiety. See, if it is having a anthraquinone nucleus in the non-sugar moiety, that will be an anthraquinone glycoside. Like, so based on the in generally, what do you mean by glycoside? Glycoside is comprised of a 𝘂𝙀𝙎 moiety plus glycone moiety, right? So non-sugar moiety and sugar moiety, these are linked by the glycosidic linkage. But this classification, second classification, will basis on the type of non-sugar moiety. Okay, if it is having an anthraquinone nucleus, that is anthraquinone glycoside. Okay, if it is having a steroidal nucleus, that is a steroidal glycoside. Like how? Okay, so we are classifying. And the third classification is based on the type of lactone ring in the steroidal nucleus. Okay, so type of the lactone ring present in the nucleus ring. Okay, so based on these two parameters, based on these three classifications, alkaloids have been classified. But I'm going to discuss about the cardiac glycosides. Okay, so chemically we have more than 8 classifications. Okay, pharmacologically we have more than 7 classifications, but based on the lactone structure, we have classified them into 2 types. Based on the classifications, based on the presence of lactone ring, we have classified them into 2 types. But do not worry, firstly we will discuss the important glycoside, that is cardiac glycoside, in this session. Okay, yes, first of all, before going to understand what is cardiac glycoside, you should know what is glycoside. Yes, so glycosides are the organic compounds. These are naturally occurring organic compounds found in the plants and animals. Yes, the glycosides can be obtained from either plants and animals, right? And now, which upon hydrolysis, they give sugar moiety and non-sugar moiety. You can see sugar moiety, that is known as glycone moiety, okay, and non-sugar moiety is known as 𝘂𝙀𝙎𝑃 moiety. Non-sugar moiety is known as 𝘂𝙀𝙎𝑃 moiety. Clear? Okay. Yes, glycosides, these are organic compounds having both sugar moiety and non-sugar moiety. So now the question is, why they need to have the 2 components or 2 things, that is a glycan and glycone. So see, ultimately we are having the secondary metabolites, or this example of secondary metabolites, mainly to produce a therapeutic action. But among these 2, which one is responsible for the therapeutic action? Only one, that is 𝘂𝙀𝙎𝙀 moiety or the sugar moiety. Okay, 𝘂𝙀𝙎𝙀 moiety or non-sugar moiety. 𝘂𝙀𝙎𝙀 moiety is responsible for the therapeutic action. But what is the role of glycone moiety? It is mainly to improve the absorption of this sugar moiety, clear? Sorry, of this non-sugar moiety, clear? So, but in generally, these glycone moieties are ends with O's. 𝙂𝙎𝙎𝙎𝙬𝙎. Okay, so let me tell you an example of glycoside, that is 𝙭. See, whatever the plant, if it is a glycoside, definitely it should comprise of two components, that is sugar moiety and non-sugar moiety. Let us consider 𝙭. Okay, 𝙭 is comprised of a sugar moiety called 𝙂𝙎𝙎𝙎𝙬𝙎. 𝙭means sugar. Okay, it is a deoxy sugar. Okay, and this 𝘂𝐿 moiety, most of the glycosides are end with a xenin part. Okay, xenin. If it is a 𝘂𝐿 moiety, it is known as xenin. Deoxyxenin, diosxenin. Okay, if it is having a suffix of xenin, undoubtedly it is 𝘂𝐿 moiety or non-sugar moiety. But the responsibility or maybe the pharmacological activity is due to the presence of this 𝘂𝐿 moiety. But what is the role of this glycon moiety? It supports the 𝘂𝐿 moiety for better absorption, okay, for better targeting to the site of action. Clear? Yes. So this will support the 𝘂𝐿 moiety. So thereby, because of the support of the glycon moiety or maybe sugar moiety, it will absorbs more. Okay, it targets more, and thereby it shows the therapeutic response. So that's the role of sugar moiety. Clear? And yes, so let us see. So 𝘂𝐿 means it is responsible for the chemical and therapeutic activity. That's what we have discussed. And also the glycon moiety will determine the water and lipid solubility. See, this will determine the water and lipid solubility for the particular 𝘂𝐿 moiety, so that it defines, it helps for the better solubility of the 𝘂𝐿 moiety. Clear? And also not only for that, for facilitate the absorption of glycoside. See, for the both glycon and 𝘂𝐿, right? In order to, okay, to facilitate the better absorption. So the absorption was decided by the glycon moiety, which means there are types of sugars are there, right? Rhamnose, galactose, lactose, right? Digitoxose. There are deoxy sugars are there. So this type of sugar will define the solubility and absorption as well. Clear? And yes, so there is another question we can expect from this two parts or two components is, okay, so glycon moiety, chemically they are known as acetals or sugar ethers. Acetals are sugar ethers. Clear? Yes, they are known as acetals or sugar ethers. So how they will act? So simply when the particular, see, so when these glycosides are enter into our body, clear? So either by the enzymatic hydrolysis or acid hydrolysis. See, let me tell you. So how these are connected? Let me show you a simple structural explanation. Okay. So in general, what we are discussing here, a glycoside, right? Glycosides. In the glycosides, what we are discussing, so the glycosides are comprising of two parts, right? So that is sugar moiety and non-sugar moiety, right? Now sugar moiety and non-sugar moiety. This non-sugar moiety is known as, or it is considered as a xenin part, and it is known as 𝑂s part, right? It is known as 𝑂s part. Clear? So now see, they are connected by a bond called, a link called a glycosidic linkage. They are connected by a link called a glycosidic linkage. Okay, this is known as glycosidic linkage. So, okay, this glycosidic linkage may be sulfur, oxygen, nitrogen, or carbon. Any of this. Clear? But okay, but the thing is that, so these together, so what we have discussed, upon hydrolysis, they yields water molecules. See, in order to break down, in order to separate into non-sugar moiety and sugar moiety, what we need to do? Hydrolysis, right? So when we subject this particular glycoside into hydrolysis, which means breakdown or removal of the water molecule. So OH group in the sugar moiety and H group in the, or H atom in the non-sugar moiety. So both will be removed as a water molecule. Okay, both will be removed as a water molecule. So thereby they'll separate together, and this non-sugar moiety will directly go and enters to the, or it is attached to the site of action, and thereby this sugar moiety will support this non-sugar moiety for the better absorption and solubility. Clear? See, ultimately upon hydrolysis they lose water molecule. So which means this OH and H will be the, okay, a bond, or maybe this molecule will, will hold this both a glycon and glycon moiety. Clear? Yes. So let me go to the slides. See, what they need to do after, see, this will be happening when this glycosides enters into the systemic circulation. They need to be dissociated into the sugar and non-sugar moiety. Clear? So then only this non-sugar moiety it go and sticks to the walls of the particular site of target. Clear? So that's the reason they should be hydrolyzed. Okay, they should be subjected to the hydrolyzed, so that they can be dissociated. Into the sugar moiety and non-sugar moiety. Clear? Yes. Okay. Yes. So that's what we have discussed now. Chemically, they are esters or sugar ethers formed by the interaction of the hydroxyl group of sugar and non-sugar moiety with loss of water molecule. That's what we have discussed. That's what I discussed now. Clear? Yes. Now, so we are discussing here in this session about the cardiac glycosides, So you know very well that the GDC is most famous for the short tricks and mnemonics and for the smart study. So we used to simplify the subject or concepts into interesting stories, mnemonics and short tricks. Within this image, see, believe me, within this image, we are going to complete the entire chemistry, okay, the chemical test and also the structures of the cardiac glycoside. Okay, won't you believe? Just go through the line, just go through the funny short trick or funny case study which we have prepared to understand the nucleus of the cardiac glycoside and also their identification test as well. So within this line we have completed the entire chemistry and the chemical test as well. Okay, so let us go for the simple case study. In the cardiology lab, there is a cardiology lab. In that, one person named Hill, he found the sugar, and then the lactone ring crew. There is a crew which they are specially for the identification of lactone ring. Okay, so the lactone ring crew, they are nothing but 𝙂𝙂𝙎𝙬𝙁, Raymond and 𝘀𝙬𝙂. So these people name called 𝙂𝙂𝙬𝙁, Raymond, 𝘀𝙬𝙂. These are the lactone ring crew. Their work is to identify the lactone ring, to determine the lactone ring. Okay, and also they have confirmed the structure of lactone ring. While there is another two people called steroidal crew. Okay, so they are nothing but 𝘂𝙂𝙎 and 𝙂𝙂𝙎 and 𝘂𝙀𝙁𝙁. Okay, 𝘂𝙂𝙎 and 𝙂𝙂𝙎 and 𝘂𝙀𝙁𝙁, and they confirm the lactone ring in the steroid. See it. So there is a cardiology lab in that a person identify the sugar moiety. First they identify the sugar moiety. Then after that, okay. So then after that, let me highlight the things so that you understand more. Okay. So in the cardiology lab, which means these are cardiac glycosides, right? These are the cardiac glycosides. Okay. So there is a cardiology lab. Okay. So in the cardiology, there is a person called Kill. His name is Kill, and he identified the sugar. Okay, and yes, there is another crew called lactone ring crew. They are nothing but a lactone ring crew. Okay, lactone ring crew. So nothing but 𝙂𝙂𝙎𝙬𝙁, Raymond and 𝘀𝙬𝙂. These people were the mainly to determine the lactone ring. Okay, and also they have confirmed the structure. So there is a presence of lactone ring. Then while there is another team called steroidal team. Okay, so they are namely 𝘂𝙂𝙎, 𝙂𝙂𝙎 and 𝘂𝙀𝙁𝙁. So these people were to confirm the steroidal nucleus. Okay, so now within this test we have completed the chemical structure and also the identification test for the cardiac glycosides. Clear? So let me tell you. Okay, so killer 𝘂𝙀𝙀 is to identify the deoxy sugar. Killer 𝘂𝙀𝙀 is to identify the deoxy sugar, and also this 𝙂𝙂𝙎𝙬𝙁, Raymond, 𝘀𝙬𝙁 tests, they are mainly to confirm the lactone ring. Here, so these tests are used to confirm the presence of the lactone ring. Okay, and yes, so another test, the remaining test, nothing but 𝘂𝙂𝙎 test, 𝙂𝙂𝙎 and 𝘂𝙀𝙁 test is mainly to confirm the steroidal nucleus. Clear? So killer 𝘂𝙀𝙀 test is for to confirm the deoxy sugars. Clear? And yes, 𝘂𝙂𝙎 test, 𝙂𝙂𝙎𝙬𝙁, Raymond and 𝘀𝙬𝙂, they are mainly for the confirmation of the lactone ring test. Okay, and also 𝘂𝙂𝙎, 𝙂𝙂𝙎 and 𝘂𝙀𝙁𝙁 test is mainly for the identification of the steroidal nucleus. See, so within this image also we know the basic components of or essential components cardiac glycosides. Yes, so steroidal nucleus should be there. Okay, so to be a steroidal glycoside or cardiac glycoside, what it contains mainly? First, it contains steroidal nucleus. It is mainly for the therapeutic activity. Steroidal nucleus is mainly for the therapeutic activity. So after that, what it should have? It should have a specific lactone ring. Okay, so that's the reason lactone ring is there. Okay, so the lactone ring is mainly for the targeting. It is nothing but a chromophore or colored compound. Okay, so it is mainly for importing of the targeting, and mainly as it is a glycoside, it should contain another part called a glycone part. Okay, glycone part is mainly for the to determine the water solubility and lipid solubility. Clear? So now we can see the basic components of the cardiac glycoside is a glycone moiety, that is steroidal and non-lactone ring comes under the glycone part, and the remaining one part is nothing but glycone moiety. Glycone moiety is nothing but the deoxy sugar. Clear? So within this one image, you completed the identification test and as well as the basic essential components or the basic structures of the cardiac glycoside. Clear? So Killer-Kinase test is for to identify the deoxy sugar, and yes, lactone crew, that is 𝙂𝙂𝙎𝙬, Raymonds and Kedis for the lactone ring confirmation. Okay, and 𝙂𝙂𝙎𝙬𝙀 and Pett-Cofer test is mainly for the confirmation of steroidal nucleus. Okay, so first, as it is a steroidal glycoside, it should have a steroidal nucleus. That steroidal nucleus should have a lactone ring, and both are should be connected to the deoxy sugar or sugar moiety. So that we have represented here. A glycone moiety, which means a glycone moiety should contain steroidal lactone ring. Okay, and as well as it should contain a glycone moiety, that is deoxy sugar. Okay, so let us see more in detail about cardiac glycosides, but such type of shortcuts will help you, okay, very easily to remember these things. Now let us see more about the steroidal glycosides. First one, a glycone moiety. See, it contains two types of glycone moieties based on the lactone ring. Based on the lactone ring, we have confirmed. Okay, so based on the lactone ring, we have classified them into two types. One is the cardinolides and the one is bufordinolides. Cardinolides and bufordinolides, but both are having same property, that is cardiac property, cardiac action. Clear? And yes, so but chemically this structure of chemical cardiac glycoside is known as cyclopentanoperhydrophenanthrin. They'll ask you this one, cyclopentanoperhydrophenanthrin, which means as they are steroidal glycosides, they are having 3 cyclohexane rings and 1 cyclopentane ring. 3 cyclohexane, which means 6 membered rings are 3, right? Okay, 3 benzene rings, okay, 1 5-membered ring. Clear? Cyclohexane 6, okay, 3 benzene rings with 1 cyclopentane 5-membered ring. Okay, 6-membered ring, benzene and 5-membered ring. Clear? So that's a steroidal nucleus. That's the reason they are known as cyclopentanoperhydrophenanthrin nucleus. They are known as cyclopentanoperhydrophenanthrin nucleus containing compounds. But so what makes a difference for this cardiac glycosides and the bufordinolides? See, do you believe that within one letter we can differentiate, within one alphabet we are able to distinguish whether it is a cardiac glycoside or cardinolide or bufordinolide? So do remember an alphabet called B. So what is a heterocyclic ring? Starts with B. B for benzene. Benzene comprises how many rings? 6. Okay, it is a 6-membered ring. Which means bufordinolides are having a 6-membered ring as a lactone ring. Bufordinolides are having a 6-membered ring as a lactone ring. Okay, but whereas cardinolides are having 5-membered ring as their lactone ring. So let us see the example. Thereby I'll show the structures. Thereby you'll remember more easily. Cardinolides are having C23 steroidal having 5-membered unsaturated ring. Okay, unsaturated 5-membered ring. I'll show you the image. Okay, so you can see here itself. Okay, 5-membered unsaturated ring, that is cardinolides. Okay, 6-membered unsaturated ring, that means okay bufordinolides. Okay, 6-membered, B for bufordinolides, B for benzene. Benzene having 6-membered ring. Thereby you should remember. Clear? Yes. So that's the reason you can see here itself. Okay, cardinolides are having C23 because 5-membered ring at C17 beta position, C17 beta position. Okay, if it is having 5-membered ring, that is known as cardinolide. Okay, and whereas at C17 beta position it is having 6-membered ring, that is bufordinolides. Okay, do remember this one. Thereby you can remember easily the other one as well. Examples are like cardinolides are digitalis, thivita and supanthus. For bufadienites, okay, just do remember, okay, squill is the best example, squill, okay, and as well as bufotoxin, toadfish, okay. So these are the best example. But the thing is that to produce the maximum activity, to produce the maximum cardiotonic activity, they should be attached, the lactone ring should be attached at a position that is C17 beta position or 17 beta position. 17 beta position, you can see here itself, okay, 17 beta position, okay, yes, and the sugar should be attached at a 3 beta position, 3 beta position. That's the thing you should remember. Clear? Yes. And now, so if you want me to explain more about the cardinalites and bufadienites, so these are the two components. Okay, so where the cardinalites contain 5-membered ring, 5-membered ring, that is alpha beta unsaturated ring at the C17 position. But whereas in the same thing, okay, 6-membered ring, that is doubly unsaturated ring at C17 position. Okay, 6-membered ring means benzene means bufadienites or bufadienites. Clear? Examples are digitoxin, digoxin, zitoxin, 𝙂𝙂𝙏 and convolatoxin. These are the cardinalites, more of cardiotonic activity. So both are having the cardiotonic activity, but more of these these cardinalites are having more cardiotonic property. Bufadienites, nothing but bufalin, silarin, which means squill, Indian squill, right? So these are best example for the bufotoxin or bufadienites. Okay. Yes, when you see the sources, digitoxin is obtained from the digitalis, strophanthin, okay, convolvulus area, okay. But whereas, do remember this one, bufotoxins, they are more of toxins, they are more of poisonous nature, they are more toxin and more potent. Bufotoxins, okay. That's the reason, mostly for the cardiotonic property, for the cardiac treatment, people will rely on this cardinalites only, for the cardinalites only. Clear? Because as these are more potent and toxic, okay, these are more preferable cardinalites, okay, these are very potent, so the handling will be somewhat difficult, and also they are very toxic. Clear? Example is squill, toad venom, 𝙂𝙁, okay, squill, toad venom and 𝙂𝙁, so they are more of toxic and more potent as well. Clear? And yes, so they're showing the moderate activity. That's the reason mostly the people will rely on the cardinalites. Okay, examples, mostly they found in plants only. Especially they are only found in plants, cardinalites, okay, but whereas bufadienites can be found in both animals and the toadfish. Clear? Yes. So this is the difference between the cardinalites and bufadienites, and you can see the structural difference between the cardinalites and bufadienites. But okay, but these glycosides, especially, okay, C glycosides, okay, based on the glycoside linkage, we have classified them into C glycosides, O glycosides, S glycosides and N glycosides. But C glycosides are not easily hydrolyzable, but they can be hydrolyzable with the oxidative hydrolysis, in which we use FeCl3 to hydrolyze them properly, Clear? And also, we already discussed that, okay, the steroidal nucleus is responsible, do remember, because of the steroidal nucleus lactone ring only the cardiac activity has been produced. That what we have seen in the case study of cardioglycosides as well, right? Sugar moiety, lactone ring and steroidal ring. Clear? So which means the steroidal lactone ring with the sugar moiety is nothing but a cardiac glycoside or steroidal glycoside. Clear? And yes, whereas if you see the distribution or biological sources in which families they can abundantly found. So here itself they are Leguminosae family, Stridulaceae family, Cruciferae, Scropularaceae, nothing but example is Distalis, okay, U4BACA. So in these species, in these families, we can find the cardinalites, but whereas in the bufadienites can be found only in the Liliaceae and Ranonucleaceae family. Okay, they are very limited. Clear? Yes. So now I'm moving to the very important test. That's what we have already seen in the case study, right? So case study of cardiac glycosides. See, so please kindly go through the image, thereby you'll understand more about the identification test and structure of cardiac glycosides. Okay, first test, Killer-Killarney test, we already discussed. Killer-Killarney test is mainly to identify the sugars, which means deoxy sugars, okay, like digitoxo sugar. Okay, mainly it is for the digitalis, especially for to identify the digitoxo sugar. So thereby we can confirm it is an cardiac glycoside. Clear? So where we have performed the extract with the plant extract with the glacial acetic acid and FeCl3 and concentrated H2SO4 upon So they give the blue or green ring at the junction. Thereby you can confirm. Okay. And yes, legal test upon its a pyridine solution and nitroprusside solution and NaOH, they gives a red pink color. 𝘖𝙎 test gives a orange color with a section of distylis and sodium picriate solution. And Raymond test will gives you the violet color with a C2H5OH 50% of the methanol and ethanol and NaOH. It gives a violet color, and Kedis test with the Kedis reagent they gives a blue or violet color. Okay, and lastly antimony trichloride test they gives a blue color with the antimony trichloride test. So don't worry, they won't ask you about the composition of the chemical test or the reagents about the chemical test. But so what you need to remember is, you need to remember, okay, so which plant is identified by which test. So they are focusing on which thing. So 𝘂𝙬𝙎 test is mainly to identify the deoxy sugar. So these things you should remember. No need to remember more about the reagents, but you need to remember more about this one. Okay. So 𝘂𝙬𝙎 test is mainly to remember the deoxy sugar. Okay. So for that we have given you the shortcut, that is 𝘂𝙬𝙎 sugar. So 𝘂 eats more sugar. So which means 𝘂𝙬𝙎 test is to identify the deoxy sugar. Okay. So thereby they gives a blue or green color. Okay. And yes, to Baljet, Raymond, Legal and Keddy, these are the lactone crew already we have seen in the shortcut, right? So Raymond ring is legal in Keddy city, are the shortcut we have created. Okay. So there is a ring called Raymond ring. Okay. Is legal in the Keddy city. Do remember, Raymond ring is legal in the Keddy city. Okay, Raymond, legal, and Keddy's and Baljet, these are identification tests to identify the lactone ring. Unsaturated lactone ring can be identified by this test. Okay, so orange or red purple color is observed. Okay, next, Lieberman, Buchert and 𝙂𝙀 test. These are to identify the steroidal nucleus. Already we have discussed, these are the steroid crew or steroid team. Right. So Lieberman and 𝙂𝙀 confirms steroids. Right. So thereby they gives a green or red color. And yes, lastly, so sugar portion can also be identified by the Pettenkofer test. So they can identify the steroidal sugar actually. Okay, so the sugar, they identify the sugar which is more connected to the steroidal nucleus. Okay, so they gives a red color. So these are the important tests and the part they were detected in the cardiac glycosides identification test. Now I'm moving to the important thing. Okay, the drugs which are very, very important, especially with respect to the cardiac glycosides. So in this also we need to discuss about, okay, the synonym, biological source and their family, chemical class, chemical constituents, microscopic feature if needed, okay, and identification test, uses and adulterants. So these are the things we need to discuss. Let me start with the synonym. This style is synonym is known as foxglove leaves. It is a very, very important because their similarity and their structure, their morphological similarity will be a foxglove leaf. Okay, yes. So biological source, it is obtained from the Digitalis purpurea. Okay, so you're able to identify the chemical constants if you have watched my video which we have uploaded in our YouTube channel. Okay, so mastering the phytoconstants, we have uploaded the video. Please go and watch. If not, okay, please go and watch. Digitalis purpurea, digitoxin. Yeah, and also from other sources like Digitalis lanata and digoxin. Digitalis lanata and digoxin. Clear? And belong to family. Okay, Scroplaraceae, but now it is considered as a Plantageneaceae. Now it is called as a Plantageneaceae. Clear? And this classification is cardinalites, which means it is a 5-member lactone ring you should have. Okay, chemical constants are nothing but purpurea glycosides A. Okay, A and B. Now they are calling as a digitoxin and digoxin and Gtoxin. Okay, purpurea glycosides A and B. Later on that, okay, secondary we have digitoxin and Gtoxin. Clear? And yes, so microscopic features, main features we need to discuss, especially they contain, okay, non-glandular trichomes, and also anamocytic stomata will be there. Anamocytic stomata will be there, and there are no calcium oxalate crystals. And we know already 𝘂𝙬𝙎 test is mainly to confirm the digitalis because it contains digitoxo sugar, that is a deoxy sugar. Clear? Yes. And also we can confirm based on the legal test as well as Baljet test. And uses, it is a cardiotonic, mostly used in the CHF, okay, and cumulative effect as well. So adulterants are nothing but mullein leaves and primrose leaf. Do remember these two, they are very important. Mullein leaves and primrose leaves. Mullein leaves and primrose leaf. Clear? Yes, now moving to the next drug, that is Indian squill and European squill and strophanthus. These two are very, very important, especially for the squill. Synonym is very, very important. Okay, sea onion or zangali pyas. Sea onion or zangali pyas or silla. Silla is a synonym of squill. Clear? So zangali pyas is synonym for Indian squill. Pyas means onion, right? Zangali means wild onion or sea onion, you can say. Clear? And yes. So whereas let us go for the squill firstly. Bulbous hose, it is obtained from the Ardenia indica. India indica. So they'll ask you which of the following is a biological name of Indian squill? Ardenia indica. Okay, Ardenia indica and Ardenia martima, that is European or white squill. So it is belong to the family Liliaceae, but now it is called as a Asparagaceae. Formerly it is known as Liliaceae family. Clear? So either you can see Asparagaceae or Liliaceae. Clear? Yes. And now chemical classes, it is a bufferdanolide, which means squill is one of the potent and toxic cardiotonic. Okay, it belongs to the bufferdanolides, which means 6-member ring is there. Okay, so chemical ones are bufferdanolides like sillarin, because the synonym is sillarin A, that's a major active component. Also prosillarin A. These are the active constants of the particular squill. Okay, but it has a, what is the characteristic feature? What is a characteristic microscopic feature of the squill? It contains a needle-shaped calcium oxide crystals. Needle-shaped calcium oxide crystals. That is a one characteristic feature. Okay, and also it contains the vascular bundles are the vascular bundles with annular and spiral xylem vessels. Annular and spiral xylem vessels. So these are the microscopic features, and followed by the identification test. We know already. Okay, Baljet test. Okay, gives a purple and red color, reddish color. Okay, purple and reddish color. Clear? And yes. So uses mainly they are cardiotonic, more of expectorant. Okay, so expectorant also we are using for squill, we are using for the expectorant. Clear? And majorly rodenticide. Squill is majorly used as a rodenticide as it is belong to the bufferdanolides. Bufferdanolides are more toxic and more potent. So to kill the rodents, it is used as a rodenticide. Okay, this application is very, very important. Do remember, it uses a rodenticide. Okay, yes, adults are Silla indica, Ardenia indica, Silla indica, or red squill. So that's about squill, and now moving to the strophanthus. Okay, now moving to the strophanthus. Okay, strophanthus, it obtained from the strophanthus seeds. It is synonymous strophanthus seeds or strophanthus seeds or 𝘾𝙀𝙉. Okay, and yes, it is obtained from the dried ripened seeds of strophanthus 𝘾𝙀𝙉 or 𝘂𝙀𝙉. Strophanthus 𝘾𝙀𝙉 or 𝘂𝙀𝙉 are the source. Okay, and belong to the family Apocynaceae, but it is belong to the cardinal classification strophanthus. Okay, yes, and it contains a K-strophantoside and K-stropanthin and 𝘂𝙂𝑃𝑔. K-stropanthin and 𝘂𝙂𝑃𝑔. These are the important chemical constants of the strophanthus. Okay, but it contains a special microscopic feature to identify is silky trichomes are there. Trichomes are there. Clear? And yes, but it contains it can be identified by using a legal test. Using legal test. It is mainly cardiotonic. Okay, mainly cardiotonic. But K and G stropanthins are used. But it is only used in case of emergency. But mostly people will rely on the distalis. Okay, that is a good cardiotonic. Okay, yes. And next is seeds of strophanthus seeds such as fewer hairs are used to adulterate this one. So that's about these two drugs. And moving to the next last two drugs, that is 𝘂𝐵 and 𝘂𝐵. 𝘂𝐵 and 𝘂𝐵 are the two important cardiac glycosides, especially the steroidal glycosides. Okay, let us see. So their source is mainly obtained from the seeds and leaves of 𝘂𝐵 peruviana. Okay, and it belongs to the family Apocynaceae, and it is also belong to the cardiac glycosides, but it contains 𝘂𝐵 A and B, peruviana, and neryfolin. So these are the active constants of 𝘂𝐵, and obviously it is a cardiotonic, but it is a very toxic. But more of all the cardiac glycosides, okay, all the cardiac glycosides having some activity called purgative and emetic property. But used clinically in isolated form for cardiac insufficiency. Okay, only isolated form of the 𝘂𝐵 is only used for the cardiotonic property. Yeah, and yes, So obviously, do remember this function. This function, that is, it inhibits the sodium and potassium ATPase channel, so thereby it increases the muscle contraction. So by inhibiting this one, by inhibiting this particular enzyme, so okay, sodium and potassium ATPase enzyme, what will happen? It increases the potassium channels inside the heart, and thereby it increases the heart contraction, thereby positive inotropic effect will be caused. Clear? Yes. So thereby it increases the muscle contraction. So perivoside can be perivoside as a substitute, though principal active compound. So perivoside is used for the adulterin. Okay, that's about the Peruvian, or sorry, 𝘂𝙬. Next, lastly, 𝘂𝙂𝘏, or it is known as chemical, it is known as 𝘂𝙂𝘎𝑆tropanthenine. 𝘂𝙂𝙏 or 𝘂𝙂𝘎stropanthenine. So it is also obtained from the Strophanthus gradus, especially from the seeds. See, 𝘂𝙂𝘏 is a one of the important chemical compounds which can be obtained from two species, that is Strophanthus gradus, especially from the seeds. Okay, Acanthera 𝘂𝙂𝘏, that is from the wood. Okay, it is also same family, Apocynaceae. It is also same cardiac glycoside. It is the main active constituent 𝘂𝙂𝙏. Okay, 𝘂𝙂𝙏 is the main active constituent. So it contains a rhamnose sugar, and 𝘂𝙂𝘏 is xanine. Xanine means non-sugar moiety, 𝑂s means sugar moiety. Okay, cardiotic. Okay, and the function is mainly to inhibit the sodium and potassium ACE inhibitor. That is a biochemical tool. And lastly, it can be adulterated with the other Strophanthus glycosides. Clear? Yes. So this is for the today's thunderstorm series, especially for the cardiac glycosides. But get ready for the last 10 important questions or important MCQs based on the previous year papers. Okay. Yes. So now let's start with the first question. Please do answer quickly. First question. Okay, so which component of the cardiac glycoside is primarily responsible for the therapeutic activity and chemical property? Option A, glycone moiety, B, 𝘂𝙧𝙎 moiety, and C, hydroxyl group at C14, and D, the methyl group at C14. Okay, so what is the right answer? Which compound of cardiac glycoside is primarily responsible for the therapeutic activity? A glycone, or the hydroxyl group or methyl group? No, undoubtedly the right answer will be, comment down your answer. We will be reading your comments. A glycone moiety. Okay, so because glycone moiety is not right answer, because it provides a solubility and absorption. Okay, so no hydroxyl group and no methyl group, only we are having, okay, C17 beta position, lactone ring is responsible. Okay, yes, very good. Next question. The basic structure present in the cardiac glycosides is chemically known as, the chemical structure of the cardiac glycoside is chemically known as cyclopentanoperhydrophenanthrene, pyranone, phenanthrene, and coumarin derivative. What is the right answer? Please, okay, let us know in the comment box. Let us know in the comment box. Okay, yes. So the right answer will be, okay, yes, undoubtedly the right answer will be option A, cyclopentanoperhydrophenanthrene ring is present, or chemically it is known as, that's one. Next question. Which of the following identification tests is used to specifically to detect the presence of deoxysugar? Deoxysugar. Okay, sugar means only one test is there. Okay, especially to identify the deoxysugars. Okay, by the Pettenkofer test also we can identify the sugar, but mostly more of the deoxysugar can be identified by only one test called, okay, that detective. Okay, that detective is nothing but Killer-Killarney test. Killer-Killarney test is to identify the deoxysugar. Okay. Next is, the buforanolide class of cardiac glycosides is considered to be the more potent and more toxic than cardinalides. Now, tell me which is the example. Okay, what is the example of buforanolide plant? Buforanolide, digoxin, 𝘂𝙂𝙏, silarin, or convolatoxin? What is the right answer? So undoubtedly the silarin, yes, quill is a more potent and more toxic. Okay, convolarin, 𝘂𝙂𝙏, and digoxin are more cardinalides, they are cardinalides. But where the quill or silarin is a toxic and potent buforanolides. Okay, yes. Next question. Legal test is used to identify cardiac glycosides by detecting which of the following? Unsaturated lactone ring, presence of deoxysugar, steroidal nucleus, carbohydrate portion. So, in order to understand, in order to answer this question, so you need to remember the case study, right? Who is a lactone ring crew? Who is a steroid team? Okay. So now tell me what is the right answer. Clear? Legal test. Undoubtedly, the right answer will be unsaturated lactone ring, right? So these are the lactone ring crew. Okay, 𝙂𝙂𝙎𝙬𝙏𝙀𝙍. Okay, clear? Yes. Next question. For maximum activity, for maximum cardiac activity, the lactone ring is added to steroidal nucleus at which position? Okay, C19 alpha position, C3 beta position, C17 beta position, C14 alpha position. What is the right answer? Let me know in the comment section. Please, dear aspirants, we are looking for your comments so that we know how many serious aspirants are there. Okay, yes, the right answer will be yes, C17 beta position. Okay, so we already discussed. Okay, if the C17 beta position having a 5-member ring, that is a cardinalite. Okay, and if it is contain a 6-member ring, that is buffonalite. Already we have discussed. Okay, this position, this will define and this increase activity, cardiotonic activity. Clear? Yes. Next question. The primary role of glycone portion in the cardiac glycoside is to determine the water and lipid solubility, acts as a chromophore identification test, and undergoes for the oxidative hydrolysis with FeCl3 and provide the positive inotropic effect. So what is the role of the glycone part? We already discussed. Okay, so the right answer will be option A. It determines water and lipid solubility. Okay, so thereby it provide the potency. Okay, yes. So next question, the color reaction produced by the ค์test, which means using ค์reagent on cardiac glycoside extract is blue or violet color. Yes, obviously they have explained the test, but what confirm the presence of? So by its color change, what we can confirm? Either mucilage or unsaturated lactone ring or deoxy sugar or steroidal nucleus. Again another question. So please try to remember that right case study of the cardiac glycoside. So thereby you are able to identify which test is for the which type of chemical component. Okay, now undoubtedly unsaturated lactone ring, ค์ring, 𝙂𝙂𝙎𝙬𝙏. So these tests are mainly two. Okay, these are lactone ring crew. Okay, yes. Last question of this series. So now please comment down all the right answers you got. The main mechanism of action by which both 𝘂𝛽 glycosides and 𝙂𝙂𝙎𝙬𝙏 exert their cardiotonic property. So how they are acting? What is their mechanism of action for the cardiotonic property? Option A, blocking the L type of calcium channels, activating beta adenosine receptors, increasing the potassium excretion from the cell, and thereby inhibiting the sodium and potassium ATPase enzyme. What is the right answer? So already in the classification in the pharmacognosy I have discussed, undoubtedly this is the right answer. Inhibiting the sodium and potassium is enzyme because the reason the cardiac glycosides are non-selectively inhibit this ATPase enzyme. Okay, so pump which leads to the increased intracellular calcium and stronger heart muscle contraction. So thereby they increase the calcium levels. So thereby they okay, so they produce a stronger heart contractions. So yes, so these are the today's thunderstorm series. I hope you enjoyed and hope you understand the cardiac glycosides. Yes, so like from these 10 questions, like more of the questions are expected in this pattern only. Okay, yes, I hope you enjoyed and I hope you understand the cardiac glycosides, and let us know how many right answers you have got from the 10 questions. Okay, thank you. Have a great day. Jai Hind.